Welcome back to Unit 3 of Acing AP Bio. In today's video, we will finish up talking about macromolecules. Proteins are essential molecular tools with a wide range of important functions. The polymers of proteins are called polypeptides. The monomers of proteins are called amino acids. Proteins have a very specific shape, unique to each type of protein, and the shape correlates with their specific function in an organism. Proteins have a wide range of diverse functions. These functions include structural support, for instance, like the protein collagen in connective tissue, transport, for example, hemoglobin, which transports oxygen in the human body, chemical signaling, like the hormone insulin, storage, cellular communication, like receptor proteins, defense, like antibodies, catalysis, which is a process that increases the rate of chemical reactions in organisms, for instance, like enzymes, and movement, like contractile proteins such as actin and myosin. Amino acids are composed of an alpha carbon, which is an asymmetric carbon, a hydrogen atom, a carboxyl group, an amino group, and an R group. The R group is specific for each amino acid, and it imparts specific properties. There are 20 amino acids, so 20 R groups. The alpha carbon, hydrogen atom, carboxyl, and amino group are known as the backbone of the amino acid. The R group is called the side chain. Amino acids form polypeptides via peptide bonds. A peptide bond is a covalent bond that forms between the carboxyl group of one amino acid and the amino group of another amino acid. The end of the amino acid with the amino group is called the N-terminus. The end of the amino acid with the carboxyl group is called the C-terminus. The properties of the amino acid is dependent on the R group. Nonpolar R groups make the amino acid hydrophobic. Polar R groups make the amino acid hydrophilic. Amino acids can also be charged, acidic, or basic. Protein conformation is a protein's unique three-dimensional shape. A protein's native conformation is that protein's shape in normal biological conditions. There are 4 levels of a protein's conformation: primary, secondary, tertiary, and quaternary. Primary structure is a linear sequence of amino acids. This sequence is unique to each protein and is dictated by DNA. One single mutation in DNA sequence can change the protein's function entirely and may have deleterious effects in the organism. For example, single-cell anemia is caused by a single DNA mutation, and it changes the shape of red blood cells. Remember, function always correlates with structure, and a protein structure is key to its function. Secondary structure involves the folding of the protein's backbone. Hydrogen bonding between the peptide bonds stabilizes the structure. There are 2 main types of secondary structures, the alpha helix and the beta pleated sheet. Tertiary structure involves the folding of protein into its 3-dimensional conformation due to bonding between R groups. This bonding could be hydrogen bonding between polar R groups, ionic bonding between charged R groups, London forces between nonpolar side chains, or covalent bonding between 2 cysteine monomers if the R group has a sulfhydryl group, leading to the formation of disulfide bridges. This bond is very strong. Quaternary structure is a structure that forms due to interactions between multiple polypeptide chains. An example would be hemoglobin, which has 4 polypeptide units. It is important to remember that the protein's environment affects its structure and function. If the protein is introduced to unfavorable conditions, it may denature or lose its unique conformation, and in turn it will lose its function. Changes in temperature, pH, or salt concentration can all lead to the denaturation of proteins. Now let's move to the final macromolecule, nucleic acids. Nucleic acids store and transmit genetic information. There are 2 types of nucleic acids, DNA and RNA. Nucleic acids also determine protein conformation, as information is transmitted from DNA to mRNA to protein. DNA determines the primary sequence of protein, or the amino acid sequence. Nucleotides are the monomers of nucleic acids. Now let's go over the differences between DNA and RNA. DNA, or deoxyribonucleic acid, codes for an organism's cellular activity. In fact, it contains information for its own replication. DNA is passed down each time cells divide. Genes consist of DNA. DNA consists of deoxyribose sugar, which is a pentose sugar. The reason it is called deoxy is because it lacks an oxygen on carbon 2 of the ring structure. DNA is double-stranded, and it has a nitrogenous base of thymine instead of uracil. Ribonucleic acid, or RNA, is involved in the synthesis of protein as mRNA or messenger RNA. RNA contains ribose sugar, which is also a pentose sugar, but it has an oxygen on the second carbon of the ring structure. RNA is single-stranded. RNA also has a nitrogenous base of uracil instead of thymine. So, if they give you a nucleic acid diagram on the AP, the best way to distinguish between DNA and RNA is to see if it's single or double-stranded and to see if it has thymine or uracil. Nucleotides are composed of a pentose sugar, a phosphate group, and a nitrogenous base. The sugar-phosphate pattern of nucleic acid is called the backbone. Nitrogenous bases can be divided into 2 groups, purines and pyrimidines. Pyrimidines include cytosine, thymine, and uracil. Purines include adenine and guanine. The base pairing rules are that adenine pairs with thymine and cytosine pairs with guanine. Cytosine and guanine form 3 hydrogen bonds. Adenine and thymine form 2 hydrogen bonds with each other. Nucleotides are joined covalently by phosphodiester linkage between the 3rd carbon on the sugar and the phosphate group. Remember that DNA always grows from the 5' end to the 3' end, as nucleotides are always added from the 3' end, or the 3rd carbon. DNA exists at the double helix, with each strand being anti-parallel to the other. That's a wrap for macromolecules. In the next unit, we will focus on the cell. See you then. For more bio content, hit subscribe.